Hello everyone, my name is Maria del Staff. I am presenting our work titled New PCA and Clustering Analysis for the FtsZ Tubulin superfamily. This work was done in the Centro Andaluz de Biología del Desarrollo, Seville, Spain, and it is part of my PhD project. In the lab, we were interested in the phylogenetic relationship between the FtsZ tubulin superfamily proteins. The FtsZ and tubulin families play a fundamental role in cell division in prokaryotic and eukaryotic cells. Most of these proteins are well characterized, and there are studies that confirm their cellular function. On the other hand, there are families with unknown function, such as R-tubulin or FtsZ-like. With the exception of FtsZ-like, all these families is characterized by having a tubulin domain at the N-terminal and a C-terminal domain that varies from family. With the techniques of sequence homology, we can establish a phylogenetic relationship between the proteins. With this study, we want to know to what extent the physicochemical properties of the sequences contain information about the proteins and what type is it. To carry out this study, we extracted more than 12,000 physicochemical characteristics of all the families with the E-Fisher tool and by dimensionality reduction using a PCA, we plot each protein in space. Because we know the domain composition of this protein and taking into account the previous grouping with respect to the phylogenetic tree, we color each protein according to the family to which it belongs. At a simple view, the proteins are arranged in 4 groups. From left to right, first, a heterogeneous group that includes bacterial FtsZ and archaea FtsZ1 and FtsZ2. The second group in grid formed by ZZ. The third in pink is formed by FtsZ-like, and the last group is composed of eukaryotic tubulins and tubulins-like. Then, using unsupervised learning algorithms, we test several clustering techniques to see if they can group the different families using only the physicochemical features extracted with E-Fisher. We compared the results obtained in the different clustering analyses with a PCA plot, and we found that all algorithms cluster separately those families that present domain difference in their sequences, separating FtsZ family, ZZ, FtsZ-like, and tubulins. But they are not so efficient in finding those families that present the same domain composition, for example, prokaryotic FtsZ. By experimental validation, we know that the different families FtsZ, FtsZ1, and FtsZ2 that are part of the group on the left perform the same function, but FtsZ in bacteria and FtsZ1 and FtsZ2 in archaea. In conclusion, the physicochemical features of the proteins allow grouping the proteins according to the domains present in them, even if this information was not used during training. But it does not establish clear differences between families sharing the same domain composition. Thank you for your attention.